So it's my pleasure today to welcome Professor Krishna Jagadish from Texas Tech University, who's going to present a seminar on transforming grain sorghum's yield potential through trait-based ideotype breeding. I'll give a formal introduction to Krishna in just a minute. For those of you who don't know who I am, and that may be some of you because I've been hiding out in Hartley Teakle for a couple of years. I'm Graham Hammer from the Centre for Crop Science, and my pleasure today to introduce Krishna. But first, the acknowledgement of country. The University of Queensland acknowledges the traditional owners and their custodianships of the lands on which we meet. And here that's the Jugara and Turrbal people. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. So just a reminder, we've got a 1-hour seminar, so I've asked Krishna to talk for around 40 minutes or so, and then some time for questions at the end. And if you're participating online, if you want to add questions, please put them into the Q&A tab, not the chat function. Thank you. So Krishna is now at Department of Plant and Soil Science at Texas Tech, where he's the Thornton Distinguished Chair, which he took up in 2022. And prior to that, I've known him for quite some time. He completed bachelor's and master's studies in India, PhD in the UK at Reading, and was working at IRRI on rice, and I particularly was very interested in the work he was doing on high temperature tolerance in rice. And there's some pretty amazing work. That work has received a lot of acclaim. He's a very well published scientist and a very well cited scientist for that work and others. He left IRRI to move to a position at Kansas State, which actually got me interacting with him even more because he started working on sorghum. And he became very familiar to us for this physiology work that he started doing at Kansas State. And it was then from there that he moved to Texas Tech to this senior position that he now holds. So it's my pleasure to introduce Krishna to give you his presentation. Over to you, Krishna.
I do acknowledge the traditional owners and their custodianship of the lands on which we meet today and pay my respect to their ancestors and their descendants. Excellent. Thank you very much. Yeah, good. Yeah, thanks for this opportunity, Graham. Thanks for making this happen and for supporting the trip. Had some very good discussions with the other team. Traveled to Gatton, spoke with Daniel as well as Dr. Chauhan's team. So I have subsequently a few more discussions happening over the rest of the week. But if you see something very interesting over here, if you want to reach out to me, I'm here for the next 3 days or a couple of days at least. So try and catch me. Yeah. So what I'll do today is try and take you through over the last 3 years that we have done on sorghum and try to look at the trait-based ideotype breeding and what we have been able to achieve in this direction. Right? When you look at an ideotype concept, this is the concept that I picked up while I was at IRRI. And a similar concept you can also see in maize. Okay, the whole idea is to try and see how best All right, I'll continue from here if that's good. All right, so the ideotype concept is basically you try to look at different plant parts and theoretically, of course, based with some physiological understanding, you try and see what would be the best way of putting these different traits together. It could be the leaf, it could be the stem, it could be the panicle, or a combination of all of this, right? So then you try to design this ideotype in a way that that plant is going to be efficient in your way, and you try to model it. So for this, what I'm going to do today is we picked a few of these traits which have been complex, have not been previously extensively studied in sorghum. And also, if you look at it, in my opinion, there's never been an ideotype breeding in sorghum similar to what has been happening in rice and in maize. So what I'll do today is to try and connect the sort of a genome to the phenome, but then looking at the environment or the target population of environments that you're looking at, and take you through our journey on how we have addressed the stomatal density, what we're doing with the panicle size, and then also stem lodging, which is, I think, a very interesting topic, and many of you are interested as well. I'll take a little bit of the time towards the end to share with you what else we are doing in my program at Texas Tech, because what I'm going to present is going to be a part of what we do in the program. To start off with, there were 4 themes, okay? So the first one is looking at leaf angle. So if you look at it, there's a current phenotype. I'll take you through this in more detail as we go forward. The first one was trying to look at a combination of plant height and leaf angle to try and capture better radiation use efficiency. And the second one was to try and look at stomatal dynamics, whether you can actually breed for sorghum with lower and higher stomatal density depending upon your target population of environment. And the third one is source sink relationships. Many of you know that Graham and team has worked a lot on the stay green side of it, but then also trying to look at the phenotypes between the senescence and the stay green, and if there are opportunities for us to enhance translocation and the yield, but also not lead to lodging, yeah? And the fourth one is looking at the panicle architecture or the panicle size. So the first 3 themes are primarily source driven, and the fourth one is a sink, which is basically your grain or the panicle, right? A little bit more on introducing these themes. The first one, as you can see, is we try to look at an erect leaf architecture in sorghum. If you have an erect leaf architecture just similar to that of some of the hybrid rice or maize, then you start to have better radiation use efficiency, and that could be a route where you can increase your yields. That was one of the theme that we wanted to chase. And the second one was stomatal density, right? So if you have hybrids with lower stomata, they might be better adapted to drier environment. Whereas if you have higher stomata, it also gives you an opportunity for better utilization of carbon at the cost of a little bit more water. So that would be a trait that would be really working well for a favorable environment. Because in the US, sorghum is not only grown under dryland conditions, but we do have quite a few producers who grow sorghum under irrigated conditions. So then there is a favorable environment where we wanted to utilize and benefit from that opportunity, right? And the third one is trying to optimize stay green. Stay green is great under a really drought stress condition, but when you have a favorable environment, maybe having a slightly senescent sort of a phenotype might be better, because then you have better translocation and you can increase yield. So that's an opportunity that we wanted to look at. And the fourth one is the panicle size and the strength. How can you benefit by changing all of these source components, but then you don't want all the benefit to stay in the source because they need to move to the sink, right? That's where the most economical part is. But then when you change all of this and you have your hybrid and then you have a big panicle and you got high yields, but when it falls off, it's of no use, right? So we realized this, and a year and a half ago, we included lodging as another trait into this whole system of work. So this is a massive effort which funded by the United Sorghum Checkoff program. You can see there's a lot of folks across 3 or 4 different universities over here, including USDA ARS. The folks who are dealing with physiology, there's molecular people, there's molecular geneticists, there's a sorghum breeding component as well. We have a couple of computer science folks as well in the whole story, because I think the traits are so complex, it's kind of hard for us to do everything manually. So we really wanted help from the computer science folks, and that's where they actually jump in. And I'll take you through some of these aspects where they've been really, really helpful for us, and how do you actually put all of this together? Because you're trying to change all of these traits. There could be synergies, there could be trade-offs with these traits, right? So that's why we're trying to look at the crop modeling aspect as well, and try and see what would be the best combination of these traits in a way that they would give us the most benefit depending upon the environment that we are targeting. I'll take you through theme by theme as we go. So the first theme is, again, as I said, trying to look at the erect leaf phenotype, and where you can actually increase radiation capture and as a means to increase yields. So what do you see in panel A? That's your current phenotype. That's how sorghum actually looks like in the field conditions. The panel B is, of course, it's an extreme phenotype, but it's not sorghum, it's maize. We never want to get to that stage, and I don't know if you'll even get to that stage, but also maize has its own advantages because of the physical nature of the year where it grows. But then the whole idea is, can we take some of the lessons from this and try and see if we can make sorghum a little bit more erect for capturing better radiation use efficiency? I did forget to tell you that there was a 36 number which was mentioned in my previous slide here. If you look at this on the top, there is number 36. What it means is, when you are trying to look at these traits, where do you start? Because you don't know which of these lines will have these traits. So that's where 3 of the breeders from the US, Texas and Kansas, they came together. They picked those 36 lines thinking that those would have these traits of interest. And that's how we stuck to these 36 lines going forward and doing our phenotyping exercise. So on all those 36, this is what we measured. So as a leaf angle, we measured the top 4 leaves, as you can see over here. And this was how, so that's the angle that we were looking at for the leaf angle. So the larger the number, the flatter the leaf is. So the smaller the number, the more narrower it is. So you wanted to really have a phenotype which is having a narrower angle, just so that it is more erect. Apart from what we are doing, there are also some of these efforts that are going in the mutant populations, as you can see, where there is a EMS mutant population on BDX623, where they do have mutants which are erect phenotype, but then there's a lot of cleaning of the background. The head size is small, so there's a lot of those challenges that are happening. So you could consider these as 2 parallel efforts that are going together, and whoever gets to that point is always going to be advantageous. But there are parallel efforts going in this direction. What you see in this slide is for data from 2 years from about 7 different environments. The red highlighted section shows you a very strong relationship between the leaf angle, whether it is the first leaf, second leaf, third, or the fourth leaf, with the grain yield. What that says is, the flatter leaves are the ones which are having the strongest sort of an impact in the yield, okay? So because there's a positive relationship, anything that is much more flatter is giving you the highest yield advantage. It was basically our hypothesis was to try and look at an erect leaf mutant, but then if you look at the bottom line over here where 36 lines, the plant height was only 66 to 82 centimeters. So because the plant height is so small, there was no opportunity for the plant to have that plasticity in the leaf angle. And that's why we were never able to get a phenotype to test the hypothesis. But if you look at the actual hybrids, they're about 110 to 130 centimeters. What it shows is, if you want to really study erect leaf sort of a phenotype, you really have to play with plant height to give enough of flexibility for the plant to really have that sort of plasticity for moving the leaf angle. I'll come back to this towards the end of my talk on the leaf angle, but I'll move over to the next one where we have done a lot of work is looking at stomatal density. And this was work done by Dr. Bhimanali when he was my postdoc and now a faculty at Mississippi State. Stomata is a lifeline, as we all know, involved in photosynthesis, involved in plant productivity, as you know, and also with stress management, with transpiration, because it's one of the process which maintains the leaf temperature, but also the homeostasis in a plant. So absolutely very critical. But how do you really use this trait in breeding? Because these are microscopic units which are there in a leaf. And so it was a real challenge and also a complex thing for including stomatal density or an area in a breeding program. So we started off, maybe this was while I was in K-State, we started to take stomatal imprints, and we had 311 accessions from the sorghum diversity panel, which captures quite a lot of diversity genetically. We grew them in two different locations. One is in Manhattan, one is in Hayes. Hayes is a drier location. Manhattan gets more rainfall. So there were two contrasting environments. We actually took leaf surface imprints, as you can see, using a nail polish method, where basically we use both the abaxial, which is the bottom portion of the leaf, and the adaxial, which is the top part of the leaf. 3 images per slide. Totally, we were looking at 12,000 images that we had to analyze, all right? It was a massive work for us, but that was a start that we had. And this picture actually, or this figure actually shows you the first part is the manual count. So all the 12,000 images were manually counted. So it's a massive effort, but then we got that done. But then worked with the computer science folks, Doina Correga from K-State, and that was the prediction, as you can see over here. So in terms of the data, if you look at both the environment 1 and 2, either you're looking at the abaxial or the adaxial. If you look at the predicted and the manual, you get almost a perfect relationship. The model really did what we wanted it to do. So we're really happy about the way the model was doing, but most importantly, as you can see at the bottom, for actually counting the stomata took us about 150 working days, nearly about sort of 3-4 months of a student, but then we could get the same data in about a week's time. And this was really encouraging with the process, because we were not only able to identify the stomata, but we were able to, or the numbers, but also the stomatal complex area. So all of this could be done in just 7 days. And I think this was the beauty of involving some of our computer science folks in the process. What it also allowed us to do is we were able to capture the genetic diversity, so we could actually do the GWAS on this. So one of the example is on chromosome 6, where it was a very good sort of location where you had 5 linked significant SNPs over here that you see here. And using these SNPs, we could actually go through the haplotype approach and separating out the entire population. So the first of the haplotype that you see over here in the dark brown, that means that the population which had that haplotype would have lower stomatal numbers. But the one which is in the oranges towards the end over here with GGTGG sequence would have high stomatal numbers, right? This was good because then we were able to identify these haplotypes, but numbers don't say everything, right? But you want to really look at what the stomata actually does. So we identify a few of these lines having the haplotype 1 and the haplotype 2. We regrew these plants in the next season, and then we actually did the physiological analysis on these ones. And as expected, you would see with the haplotype 1, where you have lower stomatal numbers, you're saving water with a higher water use efficiency. But on the other hand, when you have haplotype 2, where you have more stomatal density, of course, you do lose water, but then you're gaining quite a lot in the carbon, right? So I think these haplotypes and these lines that are identified are really helpful, because then you could then breed specifically for environment which is more drier, or where there is an opportunity for you to gain more carbon, but at the loss of a little bit more water. As I said, US has both these environments, so I think having a different breeding objective for these locations really made sense. Oops, I think. All right. So when you actually look at this stomata with the diversity, I just wanted to let you know that the leaf area and the environment really makes a difference. So this is the second leaf from the top from the diversity, and you can see the diversity that you see in the leaf size. And this is also here in the histogram that you see with the leaf area and the number of genotypes. What it means is if you have a very small leaf, then the stomatal numbers are really going to be dense. While you have a very big leaf that you can see over here, then it's going to be diluted. It's just a dilution factor. When you're looking at the stomatal numbers, you really have to take the leaf area into consideration and normalize. Otherwise, you would be way off just looking at a very small centimeter square area of these leaves. But also when you look at the environment, right? So even apart from the environment itself, if you look at it, the abaxial density, the number of stomata, which is at the lower part of the leaf, are always going to be higher, as you might know, which is about 40%. But then also environmentally, I said to you, haze, which is a drier environment, you will see that there is a difference in the dynamics that is happening in the stomata. So the abaxial, whereas the lower density goes down, whereas the density on the top increases. Okay, so you got to keep in mind that as you study these stomata, the leaf area and the environment makes a difference, and there is a plasticity in the plant to respond to the environment as you look into this. This one is published. The tool is publicly available. This is online. So if you have, it can run on your cell phone. It can also run on your laptop if you have your own images that you can feed into it. But then at that point of time, we said, well, all these images came out of a single lab. What is going to happen if the images comes from different labs and different people are doing it, because the image quality would be very different? Is the tool going to still do the same thing that it did for us, right? So in 22, we grew some of these lines in 3 different locations and all the 36 lines in Texas Tech. And as you can see, we collected those images just like what we had done previously, led up to about 3,000 images. But this time we didn't go through the process of putting these images through a microscope. We just basically sent it to a machine learning model that we had developed. No changes made at all. And you can see that you are up to 0.90 in the relationship between the predicted and the manual. Manual just meant these are the images that were fed into the system. So the tool was doing everything. You didn't have to do anything at all at this point, which was really good, because then if you're going to 0.9 R square, it's pretty good already for the breeding people. They don't need 0.99 because they are really interested in trying to segregate the entire populations. But what is interesting is if you look at the whole process, you have only high throughput, or you made the high throughput process only after the image has been obtained, right? But you still have to go to the field and do all the nail polish method to get those images. So you're only 50% high throughput, but the first part of it is very time consuming. At the same time, in 23, the non-destructive handheld microscope was published. And so we started to take that into consideration. And I know Chaitanya here in the audience is working along in the same lines. We did extensive work on trying to identify which would be the best handheld microscope. And Mayank is my PhD student. So what we have right now is thousands of images from the sorghum diversity panel, which you can actually walk in the field with this microscope and those images will be obtained, and they could actually be transferred into a cloud where you have the algorithm for analyzing these images. What was very nice was I was a little bit concerned in the quality of the image, because if you see, these are not as great as what you would get from a nail polish method. But the algorithm was really good, and even with very little fine tuning, we are able to go up to 0.9 to 0.95 association. What that means is now a breeder who can hold this microscope, which is even cheap, I think it's $350 or $500 or something along those lines. He could go in and phenotype his 10,000 lines that he wants to do in a couple of days, and in 10 days or so, he will get his data already available. So he can make his crosses depending upon whether he wants to choose for a low stomatal number or a high stomatal number. So that's the approach we are taking to make it completely high throughput. The third one that I wanted to talk to you is the optimized stay green. And I know that stay green is an excellent trait that we use in sorghum for a whole lot, especially under drought stress conditions. But as I said, when you have a slightly favorable environment, you could still do a little bit more on the translocation for increasing your yield and not have to completely have those hybrid stay green. That was at least the hypothesis. So here is an example where you have your corn or maize and wheat, which are completely brown at the time of harvest because they're pretty much done with quite a lot of translocation. But sorghum still looks pretty even when you are about to harvest with a red head and a green background. What that means is the greenness is because of the chlorophyll. And so there's a lot more nitrogen in the leaves at that point of time. And the idea was, do we have an opportunity in sorghum for increasing the translocation for either increasing yield or the protein content by moving the nitrogen that is there in the leaves into the grain? And this is the hypothetical sort of a picture where stay green is still a really good trait under drought stress conditions, but we could still have a partially senescing hybrid when you have a favorable environment. Unfortunately, we were not able to do a whole lot on this because whenever we started these trials, there's a lot more rain in those areas. So
we were not able to capture the phenotypes which exist between the stay green and senescence a whole lot, but that also gives you an indication that in the US there are environments where you get more rainfall and there is more opportunity for us to look at a senescing sort of a hybrid. This is a slide which is slightly off topic, but I thought I'll add that in here because of the stay greenness that you see in sorghum. Because we were really interested to see how much of that nitrogen is actually locked in sorghum even at the time of harvest. So we actually took a large area in sorghum and then all the leaves, and we looked at the whole plant nitrogen content that was left in the leaves after the harvest. Okay, if you look at it with the increasing levels of nitrogen that you had applied, you will see an increasing level of nitrogen still held in these green leaves even after harvest. What that says is you have about 30 to 50 kilograms of this residual nitrogen per hectare, even after the harvest in sorghum. It's really nice because then that nitrogen is locked and then may be available to the next subsequent crop. And that's why sorghum would be a really nice crop in a rotation system. And also conceptually, the thing that we are working is, can we actually have sorghum as a cover crop by itself? And if you have a sorghum, you really do not need a cover crop. But this is an off topic, and if you're interested, I'm happy to talk to you about it. On the theme 4 is the panicle architecture of the plant size. So all the 3 other things that I was talking to you about were mostly on the source side of it, right? So mostly related to the leaf, but this one is more on the panicle size of the architecture. Architecture, we initially wanted to look at the branches, the primary and the secondary rachis branches, which became really hard, but I'll show you what we have done on the size side of it. We took the same 350 lines, sorghum association panel, 4 replicate panels per line. We imaged it both at the front and the back side of it. And we had 4 different quadrants, as you can see, for every panicle. We took images of all these quadrants and together with the quadrant itself, leaving 5 images for every panicle. So you're looking at about 14,000 images in total for analyzing. So initially, again, we looked through the machine learning approach or RCNN model. It did really good when you had very widely spread out grains, as you can imagine. But the very initial sort of a model struggled a little bit when you had dense panicles or dense seeds. We did a whole lot of refinement on this one with Joyner's team, and basically the model worked really well, even though the seeds were really dense on these quadrants. So we are really happy with how this was happening. But then we were never of the idea for taking the panicle and threshing and looking at the seed because it's really time consuming and laborious. So we just wanted to see if we can predict the number of grains on the panicle itself. That was our effort. And we did this. So if you can see, they're a little bit off, but we've been continuously refining this model. We were able to get the seed counts across all of these 350 lines, as you can see, using this approach. And the predicted versus observed was 0.85 to 0.87, which is pretty good, I would say, in not having to do anything of the laborious work of having to go through the threshing process. But we wanted to take it a little bit further and try and see if we can actually map these things right away on the field itself using a UAV, working with the K-State folks. This UAV was flown at 6 meters above the canopy, very close, and we got the images both straight down as well as at 45-degree angles on both sides. Ran number of these models with the YOLO 7, 8, and 9, I guess, as you can see in the slide over here. And the idea was to try and identify the number of panicles. And I know that Scott Chapman's team has done this a couple of years ago. And this was not very difficult to identify the number of panicles, right? So what we are after is to try and see if we can use aerial phenotyping for measuring the seed numbers and the seed size, okay? Which was very hard to do because of the resolution of the imaging. It's very hard to capture the panicle at that level of accuracy. So I'll take you a little bit away from sorghum, and this is the work that we have done on soybeans, and this is Juliana's work where we have done on soybeans. And I'll show you a video where even with, this is on soybeans where we have an RC car, where we have GoPro cameras. And if you can see, these are all the flowers that we are sort of monitoring to look at flower abortion. So the idea is, can we actually use this RC car as an approach for a ground platform for taking high resolution panicle images in sorghum, which can then become much easier for us into the tool for detecting the grain numbers and the size on the panicle which is grown in the field. That's what we would want to try and do. And if you're interested in sorghum and anything with this approach, then yeah, feel free to talk to me. This is a beautiful sort of approach. We've worked on this for 3 years, and we've been able to really track these floral numbers over time effectively. So knowing that there's different traits that we are looking at, there are different populations that we have developed. And as you can see, the breeders have developed these populations, and we are currently dealing with 3 of these populations where they have different leaf angles and also stomata, but also have a single common parent where you can then study the genetics of this. And we would want to go through and develop biparental populations with this one. So that's work in progress. I'll go to the stem lodging right now. As I said, this is something we started a year and a half ago. We use a tool which is called as a Darling tool. It's called as a device for assessing resistance for lodging in grains. We work very closely with Daniel from University of Idaho. He's a mechanical engineer and who's actually developed this tool, which actually takes into consideration the height of the plant and the force, which then leads to the stem lodging. So this is the overview of what we are doing in the case of the lodging. I don't have a whole lot of time to go through this in detail, but what it does is we're looking at not just the lodging score or the bending strength, but looking at the morphological traits as well, height on the internode, compositional traits at different points of the stem, whether it is a cellulose, hemicellulose, or lignin, and some of the anatomical features as well, and the mechanical properties here with the rind penetration resistance, basically trying to put all of this to understand the stem lodging in a much more holistic manner. That's what we are after. This gives you, again, we focused on those 36 lines because we wanted to focus all the trait measurements on those 36 diverse lines. And this is the irrigated and the rain-fed conditions. And if you look at it, there's a wide variation in terms of the bending strength. And the bending strength is lower the bending strength, they're more sensitive to lodging. The higher the bending strength, then they're more tolerant to lodging. What was interesting is that the lines that are circled here were the same ones which really stood out well, even under irrigated and the rain-fed conditions. It's pretty interesting that they were so consistent. And here in the PCA graph, if you look at it, this is 2024 and 2023, very diverse 2 years. But then these lines which were having high stem strength, which is a 1, 8, and 5, remained with high stem strength irrespective of the environment. And the ones which are the lower stems were consistently with lower strength. It's pretty nice that there was not a whole lot of a environment impact on this one. So we would actually make it breeding easier. But we also looked at the senescence of these lines, and these 3 lines which have lesser senescence and these ones which have the higher senescence. These ones which senesce a whole lot are more sensitive to lodging. This senescence was taken at 6 different time points at the grain filling stage, which is most sensitive to lodging. Basically, this is under the rain-fed condition, and this is the irrigated, and pretty similar was the response. And as I said, if you look at this relationship over here, under rain-fed condition, it's much more stronger, where you see a lot more senescence. So the greater the senescence, then your bending moment goes down, so your tolerance goes down. Under the irrigated condition, the relationship is a little bit weaker. That means to say that probably we have an opportunity for those translocation to be increased for increasing the yield before they actually lodge. And so it's pretty supportive of the hypothesis that we were after. So looking at this and the opportunity that we had with the mapping population over here, we had the NAM populations, but then among that was SC35 and RTX430. If you looked at the previous slide, SC35 was one of our lodging tolerant one. So we took this mapping population, we had 181 random inbred lines. We've also done the senescence, but also taken the lodging scores on these ones. Very initial sort of analysis from the last year. Interestingly, we got a major QTL on the chromosome 1, which a lot score of 4 nearly 5 at irrigated, but under water limited conditions goes up to 8 with a phenotypic variance explained of 18%. right? It's pretty interesting, and we are trying to chase this one to look at what is the genetic control of the stem lodging under these conditions. All right, so I'll take you back to the start of the talk. I'm not going to repeat the whole talk. Don't be concerned. A year ago, Bill, who is our counterpart, similar to your own breeders over here, David Jordan, we decided that, okay, let's say all these traits that you're studying in inbreds, you find the diversity, but what happens if the diversity runs out if you put that into a hybrid background? Then all of this work is relatively useless in a way, but you can still get publications. So then we decided to look at the hybrids themselves. So Bill had actually tested 1,200 hybrids in 21 different locations. So he had the yield and the agronomic data from all of those 21 locations. So we picked 200 experimental hybrids that he had very good sort of a feeling of how they performed across different locations. And then across these 200 experimental hybrids, we took the stomatal numbers. We have the lodging scores, plant height, leaf angle as well, just to see if these hybrids had the leaf angle that we were not able to identify the inbreds in the very first sort of a theme. If you look at it, all of these 200 hybrids, majority of them has very low scores, which are all less than 20. So I think there is a lot of opportunity for us to increase the lodging tolerance in hybrids. So we wanted to have a reference where we are in terms of the current hybrids before we start improving any of these traits. So if you look at the estimates of the hybrid performance on all of the 200, just look at the bending moment over here, which is the lodging tolerance and the leaf angle. Bending moment, all of them are less than 20. But if you look at some of the tolerant inbreds, they have a bending moment greater than 25. That means that there is an opportunity for us to use some of these inbreds for improving these hybrids. Same goes with the leaf angle across these two locations where you had the College Station and Lubbock. You're looking at about 40. Yeah, angle. I told you the higher the number, the more flatter it is. So you want a much narrower angle for leaf erectness. So you have a number of inbreds which are less than 25 degrees. So basically it tells us that we have an opportunity for improving the leaf angle as well as the lodging in the current hybrids that we have. So I think this was something interesting for us to go forward. We also have the stomatal dynamics on these 200 hybrids, both the nail polish method and the handheld microscope. And we are now currently going into the genomic prediction models with Bill's team. And I think within this year, we'll have most of this done because we have all this data in place. Probably a couple more slides before I wrap it up to give some good time for questions. Again, this is where I'm looking up to Graham, Eric, Alex, where we can actually involve AppSim team here from UQ, because when you start looking at so many traits, there is trade-offs, there's synergies. What is the level or the optimization of these traits to be more effective? What are they going to do with the different management and different environments, right? So that's where probably AppSim would be really helpful for us, and also looking at how these traits are going to perform in the coming years, right, under changing climate. So I look forward to those discussions on utilizing some of this data that we have, stomata, lodging, whatever, and try to see how we can actually better utilize AppSim. It could be a win-win where we can get outputs for the trade-off, but then also improve AppSim in the process for some of these traits. So that is a part of what I do. I do multiple other things as a part of my Texas Tech role. I also direct the crop forage livestock systems, where I look at sorghum cotton rotation with and without cover crops, end to end in terms of forages, where we have grazing as well as the silage components as well. Still do a lot of heat and drought stress work, where I'm trying to build some heat tents and trying to look at high day and high nighttime temperature stress on cotton specifically now. That's one part. The second part is where I also run the Texas Alliance for Water Conservation, where I work very extensively with the producers in the region. Look at all of these sensors. AquaSpy could be arable, autonomous pivot, or Goanna, spread across these different regions with different producers. We get all the data back from the producers, and then we are able to connect these not just to the sensor data for the water use efficiency, but also look at the GIS and the remote sensing, and then go all the way up and providing newer approaches for better water conservation, and then work with multiple teams across the department in my role as the director for the Davis College Water Center, where we are trying to have a very transdisciplinary approach for looking at big ticket ideas going forward. So these are some of the things that I thought I will just put it up in case any of you are interested with outside of the sorghum. We still do a lot of stuff outside of sorghum, so I'm happy to discuss with you all. So that's my team, basically, who do all the great work on the hard work, all the funding agencies. The sorghum work is primarily funded by the Sorghum Checkoff program. And finally, I would want to invite all of you guys to Texas, Lubbock. We have this Global Sorghum Conference happening in September 14 to 18 and 26. You're all welcome, whoever is interested in sorghum. It's going to be a vibrant sort of a meeting. So if you have any questions, Graham as well as Eric, who is in the audience, are on the scientific committee. So reach out to them. And I hope to see many of you in Lubbock in September. With that, I'll stop. And Graham, you want to take it over the question and answers?
Okay, thanks very much, Krishna. Very comprehensive presentation of all the things happening at Texas Tech. I did want to mention, I'd put this thing up the front here. This is a small short black in Texas. Okay.
Texas side, yeah.
So we've got a couple of questions on the chat, and if there are any questions in the room, please let me know. And Megan will bring a microphone to you, so you'll be heard. So maybe we can start with one of these.
Sorry, guys, I'm just going to try and adjust the microphone so you can hear from the actual mics, because I know the lapel had a bit of an issue. So while he's asking the question, I'll just be trying to make things work in the background.
Okay, there's a couple of questions in here. Is there any reason why the correlation was low between height and yield? I know in sorghums in Australia, there's quite a substantial correlation between height and yield.
Yeah.
You might need to come.
Oh, here. Yeah, that's an interesting question, and if I can answer that in a different way, it's more of an opportunity where there's a couple of publications also from Bill's team that for some reason we've always chosen shorter hybrids in sorghum for whatever reason. I don't know. It's probably for the mechanical reasons for harvesting, but there's a lot of work that is done where you could still go up by a foot and a half with the hybrids that we have without even before they can lodge and increase the yields. So this is something that we really need to work towards in a way where a combination of plant height and yield. And if you look at both rice as well as I think in corn as well, where they've used height to the advantage, but sorghum community, in my opinion, has not used the height to the advantage. We have actually reduced the height. In fact, we should be actually increasing the height somewhat to be able to gain that advantage. That's what is my sort of 2 cents. Graham, if you have.
Well, I suppose the David Jordan response might be, yeah, but then you've got to watch out for lodging.
Yeah,
so you've got this trade-off depending on the environment. So this next question, any estimate for the heritability of the stomatal density as a trait?
Oh, I don't know who. Well, I don't have the number on the top of my head, but I can send that information over to you. Just send me an email and then I will send it. It should be in the plant physiology paper, but I can also look it up and let you know. But it was pretty strong as far as I can remember. I don't really remember the number on top of my head. Sorry.
Thanks, Krishna. Any questions in the room? We got one. Okay. Char?
Well, thanks for the presentation. Just have 3 questions. So yeah, so the first one is about leaf angle and radiation use efficiency. Have you looked at the leaf curvature and how did you quantify the radiation that's intercepted by each leaves of the plant, and what's the ratio between the sunlit leaves and also the shaded leaves?
So do I need to repeat the question? So the question for those online was about leaf angle and radiation use efficiency. And just correct me if I haven't got this quite right, Joe, but how do you handle the interaction between efficiency and the extent of interception? Is that what you're
Yeah,
You're
and
giving it?
how did you quantify the radiation?
Yeah, so go ahead. Got the idea.
Yeah, if you looked at the results, it was not really motivating. So the hypothesis was we wanted to look at a erect leaf sort of a phenotype. But when we looked at the data, it felt like we never had that phenotype to really do all those measurements that we wanted to do, where depending upon what is the height and how the radiation use efficiency was intercepted, because all the 36 lines pretty much behave the same way. So there was really no motivation for us to really do more work on how it is intercepting or how much is intercepting. But maybe when we look at these 200 hybrids, and if we are able to at least get a moderate level of phenotype where we are able to get the higher part into a more erect one, then you would actually have sensors at different places along the canopy, and then you can measure the light interception. And then there is more motivation to do. It's like a non-significant response. And so if I get a phenotype which would really fit the hypothesis that we are looking for, we definitely would want to do that. But for now, we didn't have the motivation to do extra work when the phenotype didn't even sort of come up to what we were looking for. So
Yeah, great.
Go for your
And-
second question.
Yeah, and the second one is for the over 200 lime sets with different stomatal density you mentioned. Yeah. So have you compared the yields and water use efficiency of the lines with low stomatal density but with different radiation use efficiency? For example, the leaf
So
angle.
just to paraphrase the question, it's about the stomatal density and whether or not there was an attempt to compare the yield of lines with differing stomatal density and their light use efficiency. Is that the question?
yeah, I can give you a definite answer in a couple of months. The reason is the first effort that we didn't want to associate the leaf stomatal numbers to the yield was because it was mostly on inbreds. But with the 200 hybrids that we have the stomatal numbers, we have the yield across multiple locations. So what happens is if you have a single location and you start to draw conclusions with that, with low stomatal number giving you different. So also let me take a step back. So all of those data initially that I presented were from single location. So if I started to say that these bunch of ones which are having low stomata has higher low yield, and those are also not tested in different locations. So that way it would be very erroneous for us to give those sort of conclusions. So what's happening now is with the very last slides that I presented where you have the 200 hybrids, they are grown in 21 different locations. So I have the yield for all of that right now. So that's why with the genomic prediction, we will be able to associate the stomatal numbers and the lodging to the yield, whether these were in a slightly more favorable environments or in a drier environments, and then we'll be able to classify and give a better answer. So within this year, that paper should be out. So there in that paper, I'm pretty sure that we will do all of these sort of associations, keeping the yield in mind. So yeah, that's why we didn't do that. We were very cautious on not doing that.
Thanks. So the last one is you show the plots with lower stomatal density but higher intrinsic water use efficiency. Yeah, so have you looked at the water use efficiency at the plant level and how strongly the correlations between intrinsic water use efficiency and the water use efficiency at the plant or crop levels?
Okay, so I'll just repeat. The question is the association between stomatal density and water use efficiency that was indicated, and does that hold up to with water use efficiency at plant level? So I gather the water use efficiency may have been leaf level, was it?
Yeah, that was at leaf level.
Okay, so I suppose the question is, does that carry through to plant level?
Goes from the leaf conductance to leaf intrinsic and then plant level.
No, at the plant level, we don't have it. It is all at the leaf level with the intrinsic water use efficiency that we have done. For that, you'll have to do a completely set. So you take the same set of lines and do, or if you want to model it for a whole plant with an absent, that might be an easier thing to do as well, because when you have all those data, you should be able to sort of pick it up in a modeling exercise. Yeah, so whole plant, we haven't done it. Yeah, it's at the leaf level.
So we'll go, I'll do one from on here, then come back to you, Fernando. So the next question from the chat. Having variety ideotypes for certain environments is idealistic. However, how many environments do you have in the US production environment? And more importantly, how many targeted varieties are commercial companies capable of deploying economically? Basically, what is the feasible middle ground between research ideals and commercial reality? Sounds like a plant breeder.
This is David Jordan. That's like, wow, that's a loaded question for sure. Let me think on that a little bit. So with sorghum, and I can see historically, if you see, we've lost ground in sorghum, and because of the market share, right? So what's happening with a new change with sorghum, with the climate smart and the water conserving nature, that's one thing. But also there's a huge push in the nutrition side with the One Health coming in the US, where sorghum is moved beyond just the traditional growing for grain, but there's a lot of sorghum being used in the baby foods and the nutrition side of it. So there's extended markets that are being opening up. So the hope is, in my opinion, there is at least a wider spectrum in the US for sorghum. And there's, with some of the climate smart commodities grants that we are doing right now and the increased insurance that is coming for sorghum, there is going to be certainly a slightly favorable group of producers in the US. I know that most of the sorghum will still be dryland, but there is always going to be, I don't know, maybe about 30%, I would say, or 25%. There would be some of these producers who are looking for high yielding sorghum variety. So I feel like there is an opportunity, but it could come to it's a hard question to answer. It's very hard, but I feel like if an opportunity, okay, let's look at it this way. If an opportunity arises, then sorghum would be ready to take up that opportunity by knowing that you could do all of this and you have the hybrids to fit the demands. But then if you never do this to sorghum, sorghum would always be seen as a marginal crop and it will be pushed more and more into the marginal crop. And there's no way that you'll ever be able to come up with the yield potential that sorghum actually can do or deserve. So there are different ways of looking at this question depending upon the context. But I'll leave it there, but I can discuss more if you want.
Thanks. Fernando, you had a question?
A very simple one. You may not have the results yet, but what's your reflection on what's been found in other species about the relationship between leaf angle and leaf size, with more erect leaves being smaller, allowing penetration of radiation towards lower levels of the canopy, and the claim that that may influence lodging?
Sorry, I'll just repeat the question. So about the association of leaf angle and leaf size, canopy development, and how that might interact with lodging?
It's a simple question, but a very interesting one, Fernanda. Sure. So the initial concept when we looked up the leaf angle was also trying to see if we can increase the planting density in sorghum. So it's very well proven. If you have a more erect leaf, of course, there's going to be better penetration. You don't want all the leaves to be erect, but of course, like a Christmas tree, an ideal sort of for visualization. Then you have better leaf penetration from the lower leaves, and then you gain more carbon. But then our idea was if you have a slightly more erect sort of a phenotype, then the idea is to increase at least in a slightly favorable environment, increase the planting density, and which is one of the way that corn has actually doubled and tripled its yields. So that was the whole idea. And then again, the lodging is a great question. Again, stay green has a very strong relationship with lodging. So if you start to senesce a whole lot, then you do have charcoal rot infections, and then you get biotic diseases also, and increases lodging. But wherever there is, again, slightly favorable environment where stay green can still prevail a little bit. So I think you can still have a erect leaf mutant with a higher plant population, theoretically, at least. That's the idea that we wanted to. But then, as you saw, we really don't have a phenotype as of now with erect leaf sort of phenotype. And so looking at those 200 hybrids, we are trying to see if this doesn't work, then probably we'll have to go to the mutant population, as I showed you with the 620 teeth that is there, and then probably use that as our sort of a longer term approach of trying to get into this phenotype. So it's all there, but when you look at that lodging, you can still play around quite a bit with the lodging and the senescence, because lodging also is not just about the stem, it's also the leaf sheath, which is even more important if you look at the lodging side of it. And that's what we are looking at right now. So that's why there's a lot of these trade-offs that's happening with the ideotype breeding. But yeah, these questions help. Thanks.
Okay. There are a couple of more questions in the chat, but I think in terms of time, inbred parents, I think you've covered those to a degree in what you've said. I suppose I did have one question. In terms of ideotype breeding and adaptation, our work here, and I think this reflects probably that question that came from most likely David, was, we're able to identify a number of specific environment types in our target set of environments. And the optimal combination of genotypes and management is very different in each of those. And so, you've mentioned these concepts of trade-offs, but really it's almost, and I think that was the basis of that question, is that some environments at the same spot, you can be in one place, and from one year to the next, your system needs to be totally different to be optimal. So the idea of having something that you can just build as an ideal combination is probably forlorn, because from one year to the next, the answer is very different. And I'm not sure if in Texas it's the same, but I suspect it probably is. And so dealing with that, and how, I suppose we put it as a question, how does one try to deal with that? Do you just try to find a combination that blends across everything as best as possible? Or do you try to find specific combinations that would more likely favour the environment type you'll get in more often in one place than another?
Yeah.
So, and that's the way we've been thinking a bit here. So I don't know if there's any comment you'd like to make.
Yeah, no, along the same lines, if you start breeding for a specific environment in mind, let's say, but that specific environment might change the next year. So if you look at just one example with the lodging one, which actually came up in QTL chromosome 1, which is exactly at the same point, and it is weaker under irrigated conditions, but when you look at the dry land, it becomes much more stronger. Some of these regions and some of these traits which pretty much can operate across a wide spectrum of environments is what would be the first year of target, because when there is an opportunity, then you want that plant to make use. So it's like opportunistic breeding, if you want to call it as, not to be a very strict saying this is a genotype. I don't know if that's the way to deal with that, but we'll try and target traits which would be effective or a wider range of environments to give it a start. And then from there on, you add on whatever you need for that specific environment.
Okay, no, that's fair enough. Thank you. And I suppose it remains to just thank Krishna for his presentation. I think we've had a useful discussion, and thanks very much for coming. I'm sure there'll be plenty of discussions over the next few days.
Sure.
Thank you very
Excellent.
much.
Thank you.
And I'm supposed to say something about next slide, but it won't go. There it goes. So just to remind people to keep an eye out for what's happening with the seminars coming up, which will be on the web page via that code. Thanks very much.